Well, hello, everyone, and welcome to week number 6 of the Genomics in the Cloud Book Club. I'm your host, KT Picard. First of all, Happy New Year, everyone. This is our first meeting of 2021. I'd like to welcome everybody here and also our growing audience on our YouTube channel following along. Today, we move into Chapter 5, First Steps with GATK, the Genome Analysis Toolkit. It really is the heavy lifting of the book. So we'll go through our usual agenda. We have a guest speaker today who will introduce the topic to us. We'll have additional resources, and then if we have time, an open discussion. So again, today it's First Steps in GATK, and it's my pleasure to introduce our second guest speaker. We now move from cloud computing to the heart of the book, the Genome Analysis Toolkit, or GATK. We're honored to have someone whose intellectual curiosity and broad academic background in medicine and immunology can guide us through our first steps. For the past 5 years, our speaker has been with Leiden University Medical Center in the Netherlands, where he did his PhD research in malaria vaccinology. His current project involves the diversity of the immune system across multiple geographic regions. It's work that's funded by a grant from the Chan Zuckerberg Initiative. Previously, he served on the Faculty of Medicine at the University of Indonesia and also works as a general medical practitioner. He's a consistent presence at our weekly meetings. I've enjoyed his insightful questions, and I look forward to his continued participation in our book club. Our guest joins us from Indonesia today. Please welcome Dr. Mikhail Manoram.
All right, thank you very much for the generous introduction, Katie. So can you hear me well now?
Yes, you sound great.
All right, so I will share my screen. All right. So, well, unfortunately, because I only have one screen today, so I will not have my presentation in presentation mode so that I can swap windows easily. But can you see my presentation now?
Absolutely.
Yep.
Great. All right. Okay, so once again, Happy New Year, everyone. And today we will start our first book club of this year by doing a quick recap of Chapter 5, which is the first steps with GATK. So we have heard of this GATK as a passing mention. Previously, we have installed the Docker VM in the previous chapter and run the GATK command to ensure that our installation works perfectly. So what is actually GATK? So GATK stands for Genome Analysis Toolkit, and as the name implies, it is a collection of tools with a primary focus on variant discovery. So if you go to this link, I will share you the slide later. You can see a lot of user guides, questions on the forums, and even down to how to install the tools, which I find really, really helpful. So I think all of us are quite used to how well or perhaps how bad bioinformatics packages are documented. And I would say that GATK is not one of them. It's, well, so far, I think it's quite well documented. There, even with so many tools in it, but well, we'll see. So GATK has a lot of tools. So if you see what are the tools that are present in the GATK toolkit, well, that's redundant. So we just have to type GATK and / list. All right, so as mentioned earlier, I am planning to do a bit of a live coding. This is very nerve-wracking. So this is the cloud that we have created last week. And to see the Docker image that we have pulled earlier, we just have to type docker ps-a to see the list of Docker images that we have. And then, okay, so I have this GATK version 4130 that we have pulled, and we have this container ID, and I run this Docker image interactively. So if we want to load the Docker image, we should also attach our disk so that we can store the results from our Docker container. So to do that, type docker run -v. So v stands for volume. And we have to know, so what is actually the home folder that we want to attach. And to do that, we just have to type ls to see, okay, so we have two folders in our VM. We have the book and snap folder, and we want to attach this book folder, just docker run -v. And then, okay, so we want to attach this book folder from our VM to a folder home book in the container. And we want to run this container interactively with the -it. And which container are we going to run? Is this one, the one that we have pulled last week. And we want to run from this container, we want to run a bash. Okay, fingers crossed. All right, I didn't make any typo. So if everything works well, so we should have this GATK and sign here. So as a reminder, if we see the $sign here, then it means that we are working in our VM. Whereas if we have the hash sign, it means we are working in our container. All right, let's see what are the commands that we have here. So we have the GATK. We can just type GATK to see, okay, so what are the possible commands that we can use for this tool. So the very basic template would be GATK, tool name, and then the arguments for the tool. And for today, one of the tools that we are going to use quite intensively is the haplotype color, which I will show later on. So if we want to look at the tools that are present in the toolkit, we just have to type GATK and /list. Well, we have to wait for a while. And we can see there are a bunch of tools that are present in this toolkit. I don't think a lifetime is enough to learn all of this, but it's just amazing, the breadth of this tool. But I digress. So now we want to run. So now we have turned on our GCP instance, and now we want to, of course, do the nice thing, which is running the actual GATK. So, and note here that the line starts with a hash sign. So it means that we're going to run all of this command right from the container. So what we want to do today is doing a variant. We want to do a germline variant calling for insertions, indels, and SNPs using the haplotype color. So first, we have to move our directory to this one, to this germline. So, and CD stands for change directory, home, book, data, germline. And as you can see here, I already have the sandbox folder. It's because, well, previously, obviously, I have made this before to prepare for this presentation. But if you haven't done this, so you should create this sandbox folder, and the sandbox folder will be the container for all of your results. All right, and now we're going to run this haplotype caller. So before we even know what is this haplotype caller is actually doing, let's just run this and see what comes out of this. So when we want to run any tool from GATK, we have to type the GATK first and then the name of the tools. And then in Bash, if you want to make like a multi-line input, you can do so by having this backslash. And then you can continue your input in another line. I find this quite useful to organize your arguments. So for haplotype caller, it has 3 arguments. The RIO, so R stands for the reference that we're going to use, and we're going to use the ref.fasta in the ref folder, and the inputs that we want to use is the mother.bam. So the input for the haplotype caller is a BAM file, which is a sequence that has been aligned to a reference genome. And then for the O, this is the output. So this is for the name of the file of the output. All right. So for the sake of time, I will skip some steps. So I will just, instead of analyzing all of the data that we have, I will analyze just 200,000 base pairs of the data. So, and to do that, in addition to this reference input and output argument, we should also add the L argument, which stands for the interval, because for this occasion, we just want to restrict our analysis on a certain part of the genome. So for chromosome 20, base pair 10 million to 10,200,000. All right. Just type enter, and now it's running. Well, it takes longer than I thought, but well. So what we are seeing here are the progress of the program. Okay, now it's done. So as I typed before, the output of this haplotype caller is a .vcf file. So this is the variant calling files. And if we want to see, okay, so what are the variants that are called with the haplotype caller, we can download this .vcf file into the interactive genome viewer, and then see, okay, so what are the variants that are actually called for this sequence. All right, and however, to do that, note that everything is still in our VM right now, so that we can access the file from our IGV. We have to copy it to the Google Cloud bucket. And to do that, it is I would really highly recommend to open another shell, which I have already opened. So this is this is prefixed with a $sign. So it means I'm running it on the VM instead of the container. And okay. And well, I actually, oops, there's something there that I want to see. Okay, so what are actually already in my bucket, because well, previously I have moved everything to my bucket, of course. And to do that, we just have to make this gsutil command, and then afterwards we can type any Unix commands that we have usually used to navigate the folders. All right. By the way, if there are any questions or if I'm working too fast, please say so. All right. So I have everything here already, actually. So previously, I have the mother variants 200k.vcf file. And so this is the variant calling results. And to see what are the variants that are called, let us open our IGV. And well, always takes a while. So first of all, so the VCF file is only the variants that are called. However, we should also load the actual alignment file. Okay, so well, as in the book, we are using this mother.bam. So just go to the file URL, just fill this one. You don't have to fill the second one because I'm using the latest version of the IGV, and it will automatically load the index, if I'm not mistaken. Well, it. All right, and so we have the alignment file, and now we are taking the variant calling file. Just again, load from URL, copy paste our link from the Google Cloud bucket, and then OK. So we have to sign in, of course, to your own bucket. Well, fortunately, I didn't have to write anything. And now, don't be surprised that you don't see anything, because before we see anything, we should zoom into the chromosome. So to zoom into the a specific region of our genome, just go to this part here, type. So we want to go to the chromosome 20. Of course, I am looking at my notes. I don't remember all of this. So it's all in the 10th million base pair. 623. Aha, a typo here. There will always be a typo in live coding. All right, it's loading. Okay, so now we have here our chromosomes, and this is the variants that are called. So we have two variants that are called here. And here what we see are the evidence or the sequence reads that supports the variants that are called by the haplotype caller. So if we see at this right alignment, it seems that we have enough evidence, because there are a lot of reads that has this particular changes. And what are actually the changes? We can just click the red line, and we see that the reference is C, but then there is a SNP from C to T. And for the other one, we just have to click it again. So the reference is G, but then there are some evidence for 3 base insertion of T. However, if we compare these two variants, it seems a bit surprising because this one variant here, I hope that you can see my where my mouse are pointing. We can see a lot of evidence supporting, a lot of reads supporting this variant calling. Whereas for this one, it seems like we only have very few evidence here. This 1, 2, 3. So it's a bit surprising that of this difference in amount of in the quantity of evidence between these two called variants. However, when we see this kind of surprising findings, which especially for insertion, we should remember that what we are looking right now is the BEM file, which is the alignment file. And if we look into how GATK is actually working, so haplotype caller has 4 discrete steps to call a variance. So first, they identify active regions. So the point of this is simply to filter, okay, so what are parts in the genome that are interesting? So there are, so the program is looking at the BEM file and then try to align the BEM file to the reference genome and then see which parts of the genome that has this a lot of information or a lot of variations between the reference and the sequence, the short reads. And if an area passes a certain threshold, then what haplotype caller will actually do is it will focus in this interesting region of the chromosome of the genome, and then it will actually build what it called a plausible haplotypes. Okay, so what it's actually doing is that based on the sequence reads that we have and the reference genome, so what are actually the combination of plausible nucleotides of combination of possible haplotypes that may explain the data. And so what it does is that it actually, after this step, after knowing, okay, this region, there are a lot of interesting variation, it throws away all the alignment information, and then it does a local reassembly of the genome. So what we are seeing here, the BEM file is actually not what the haplotype caller are seeing. So we are seeing 2 different things here. And how can we actually see what haplotype caller are seeing? So to do that, we shouldn't use this plain BEM file, but we add this dash BEM out argument to our haplotype caller. Okay, so, all right. So, and all right, so we'll go now. We are trying to add this BEM out argument, and well, just one moment looking at my notes. All right. So, by the way, I think, oh, all right. So let's call the haplotype caller function again, using pretty much the same argument. So the reference ref, and using this alignment file for the input. And we want to put it into this sandbox folder, and then we name it mother variance snippet.debug.vcf. And is in addition to the VCF file, we also want to see the BAM file that is the result of the local reassembly of the haplotype color. And then debug.bam. And one more thing, because this is actually quite an expensive process, this reassembly. So we should just look into a very specific region of the genome. So here, the variant callings are between the 10 million and 2,000 and 10 million, 3,000. So we just analyze this very specific interval. So we are narrowing down the search area. All right, so hopefully no typo. Let's type this and see. All right. So as you see here, I have this the BAM file, and if you're doing it on your own, of course you should always copy this result BAM file into your Google Cloud bucket. But of course, I have done this before, and here I do have the mother variants.snippet.debug.bam. All right, so let us see what the IGV, what the haplotype color is actually saying when it's doing the local reassembly and then doing the variant calling. All right, so we just have to select this result BAM file from the BAM out argument. Again, very simple, just load from URL. Okay. All right. And now we have another, well, we can see obviously a different looking BAM file here compared to what we have before. So we see a colorful tracks now. So all these maybe ribbons are a single short reads. And we have what it calls artificial haplotype RG, which is a group of possible haplotypes or possible sequence that may explain the variation in this region of the genome. And if you see, so it has a color. So this red color, yellow, and green. And you can match this color. So this is the artificial haplotype. So this is actually a sequence that is taught by the haplotype caller. So this sequence is actually not really present in the data, but it's like the best possible sequence that may explain the variation in the data. And we see this red here, and this haplotype can be explained or supported by the presence of all these red ribbons here. And this this one here, this yellow track, is supported by these two reads. Okay, so now we can already see that the haplotype caller calls 3 artificial haplotypes for the possible variants for this region of the genome, and we can also see the reads that may support the this variant calling. Okay, so I think, let me see. Okay. By the way, if this is not what you're seeing, then you should also group the alignments by the read group, and then afterwards color alignments by tag, and then enter the tag HC. So because previously I have done this, then it becomes the default argument. So so that is why now I have like the reads, the artificial group by the artificial haplotypes and the short reads that we actually see in our data. Okay. So that is in a very hand-waving manner what haplotype color is actually doing. So first, it really focuses on a certain part of the genome, and then afterwards, for that particular region where there are a lot of variations, it rebuilds the graph for alignment, and then afterwards, it determines the likelihood of every reads, and afterwards, it marginalizes all these likelihoods into the likelihoods. So from the per read variance into per sample genotype. So now that we have all this haplotype, all these variants called, that is unfortunately not the end of the story, because as explained in the book, this haplotype color is designed to be very sensitive so as not to miss any signal. However, then it comes with a drawback that we may get a lot of false positive signal. And it will be, I think, discussed in the following sections. But here we're going to we're also going to see this short introduction into the quality control of the variants that are called. So in this chapter, maybe there are more quality control parameters, but in this chapter, there are two parameters that are that we can see as for the quality control checking. First one is, as it has a very obvious name, which is the quality. And so as you can see here, it has very long right tail. So there are indeed, if we see the scatter plot from between the quality with the depth, then we see that there are a lot of high quality reads with a high depth of coverage. And what it meant by a high depth of coverage, it means that there are a lot of sequence for a lot of short reads for that particular part of the genome. However, it doesn't mean that these dots are real, because as you see here, the quality is confounded by the high depth of coverage, simply because if we are only looking at the quality, it hasn't normalized. It is not yet normalized by the number of by the depth of coverage, which is why it is very important for us to see the call by depth, which is a quality but normalized by the amount of coverage available. And if we see in the book, if we are making a threshold based on this metric, it is called the hard filtering. And when we're looking at this kind of plot, sometimes it's not clear where we should actually draw the line, which one is, so where is a line that demarcates a good and bad variant calls. And because of this, because of the subjectivity, and there are so many things that has to be manually adjusted, filtering the variant calls by looking at all these metrics is not recognized as as a part of GATK best practice. So what we did so far is actually just a very tiny segment in I would say quite a long sequence to call your germline SNPs or indels, which is the haplotype caller to call the variance per sample. Of course, for this occasion, we already have the analysis ready BEM file, and then earlier we see the SNPs and indels, and we are also introduced to the concept of collateral for adding all our variant calls. And next, in the next session, we will discuss what are the possible ways to do the quality control to filter the VCF, the variant calls in a more robust manner. And we will discuss that. So what are GATK best practice? So obviously, before the quality control by simply imposing a threshold is not a good practice at all. And if we go to the GATK website, well, we can just type GATK, and it will be the first website in the Google search. Just go to the getting started, and then you can see a lot of best practices, workflows, and tutorials, either in the GCP or in the Terra. And then what we are doing right now is the germline short variant discovery for SNPs and indels. So this one. And we can, there it actually explains in a more detailed manner. So what we are actually doing for every steps and so on. So I find this quite helpful. But of course, I haven't really read word by word for all of these documentations because but anyway, so for the best practice, it is what variants that we are actually wants to call, either the germline or somatics, and then the type of the variation that we want to call, either SNPs and indels, copy number, and structural variations. And what we have discussed today is a very simple and not even a best practice version of the haplotype caller. And I think next time, for the next session, we will discuss the best practice for the germline short sequence variants. So for this one. All right, so in addition to the book, which I think is very, very helpful to prepare us for doing all this genomics in the cloud, I find this, there is a bunch of YouTube videos of GATK tutorial by staff from the Broad Institute. So this is a 4-day workshop. You can just find, you can just click the link. I will share the link later. And I think there are a 4-day workshop, and each workshop consists of 6 hours. I think there is like 1 hour break. So there are a lot that they present here. Then they also present in more in-depth what the haplotype caller is actually doing. So if you want to know more the technicalities behind all these programs, then I would really recommend going through this. So, well, we still have 20 minutes. And any questions so far?
Michal, thank you. You made that look so easy.
Yes, thank you very much.
Thanks for presenting.
Oh,
yeah, very much appreciated.
Wow.
And it looks like Dr. Van der Oorth joined us as well. Welcome, Geraldine.
Indeed, please all call me Geraldine. My last name is a bear to pronounce.
Hello. Yeah, I just was curious. Mikhail, thank you. So I really appreciate your walking us through that. I especially appreciate your discussion about artificial haplotypes, and you were really thorough in walking through the quality issues as well. I think well represented what was there in the book, and I think it shows how the book is. If you follow it step by step, it's almost foolproof. So I again thank you for taking the time to put that into great perspective for us. I'll open it up for questions and comments. We've got a few more minutes. Would love to hear people's experiences and how they found the chapter, and then we'll talk a little bit about next week and other areas that our members have questions and concerns about. So I'll pause there. Well, I can tell you, Mikhail was going through this and he had some typo issues. I have the same. And so I think it's one of those, you just kind of have to fumble through it. And I had, when I was typing out in chromosome 20, it was 10 million. And in one place I typed a period instead of a comma, and, GATK bombs out. It very nicely tells you, I don't think this is right. I also didn't pay attention to capitalization at some point, and it said, maybe you meant, and it spelled it out. So there were some kind of, clearly people have had the same problems before. And so GATK tries to catch the common ones, obviously. But I found that very helpful. Again, having the environment packaged together, I played with Picard, no relation, by the way, to my last name, but play with those tools as well. And they're now all included with GATK4, so that was a welcome addition to learn about. And Geraldine, can you tell us a little bit about, you used this genome in a bottle mother sequence. Is there a particular reason that you used that sequence for, for illustrative purposes?
Yeah, we tend to use the genome in a bottle data sets because it's public and it's widely used for things like benchmarking and testing. And so it's just a nice reference data set to use because there's so much that's known about it. And we also have a very good estimation of the quality of the data and stuff like that. So we know it's very reliable and so on. And there are additional, references that have been based on that. So it's just a very nice, it's very complete in the sense that there's even more resources available than what we use in the book that relate to that data. So it's convenient, but you could run through the same thing with other data sets if you wanted, for sure.
No, cool. That's really good. And I just want to say that by the end of chapter 5, if you didn't have the feeling that you were a bioinformaticist, you definitely have that feeling. So because it walks you through some real world examples, and you actually go through the process of kind of debugging, if you will, how the collar got to its, how it made its conclusions. And when you talk about, you go through soft clipping and you look at that, and as Mikhail showed, when you go into the, kind of the artificial haplotypes, which seem to have a really strong influence on the collar itself, that, it's trying to make this best guess, if you will, because at the end of the day, it's a probability approach that I think that gets us to where we are, right? And so you start to see visually the consensus. And in the IGV, the viewer using the colors and changing things around by tag and by group, RG, the read group, of course, it gives you insight into where things are actually being, where they're coming from. So I found that very powerful, pedagogically, I guess is the point. I'm curious, how did you get to this example? Is this, you've given a bazillion workshops. How do you come up with, this is an example that, kind of pulls us all through the knothole and gets us to a point where it's like, oh, okay, I kind of see how it works?
Yeah, I would say that it's been trial and error over those bazillion workshops. I mean, we did the very first workshop. Well, technically there was a workshop, GATK workshop in Mexico just before I was hired, but it was more presentation based. We did the first hands-on workshop years ago. I want to say, well, it would have been 2012. Anyway, and at the time it was still unified genotyper, not haplotype caller, and it was very, you know, we'll figure it out. We just run some calls and then randomly look at some calls in IGV, and from that, which was a very unstructured first pass, and we found through that session that people still were confused. They weren't sure what to look at. And from workshop to workshop, we kind of added on based on the previous one. We're like, okay, that didn't really work. People didn't really get that part. We need to find a site that shows this specifically. Definitely when haplotype caller came out, and the artificial haplotypes and all of that, at that point, that gave us actually a lot more interesting material to put into the workshops, because you have that insight into what the caller was thinking, because you see the various options. And there's additional options if you want to see other possibilities that the caller considered but did not actually emit. Like there's additional options to have troubleshooting insights. And that just gave us, we found a lot of power to kind of expose the inner workings and explain. And I think we ended up on those particular sites. I have a vague memory of being on a train and reviewing hundreds of sites. I had written a little script to identify places where the call was different before and after, realignments based on call from unified genotyper and the haplotype caller, and then just reviewing, I think, some hundreds of sites just to find something that would be kind of compelling and that we could have a lot of things to say about. There were a lot of other candidates, but yeah, a lot of trial and error and just kind of figuring out what worked for people. So I would say a huge thank you to all the people who came to previous workshops and you know, sat through early versions, and I'd like to think it helped them already somewhat, but everybody's feedback and experiences at workshops is really what helped us refine the examples that we use now in workshops, and which were the basis for that chapter of the book as well.
Great, thank you. And you brought up a good point, which was around best practices workflow. And as you mentioned in the book, it changes over time. Mm-hmm. So if someone hands you, as you say, you know, hands you something and says, this is best practices workflow, well, best practices in 2012 is probably a little different than it is today, right?
Yeah, absolutely. So I definitely encourage people, if when you're writing up your paper or your protocol or whatever, it is simply not enough to say, I use GATK best practices, because that's vague to the point of being meaningless. And so we really encourage people to definitely say what version of GATK was used. And if you're saying GATK best practices, I think it's important because we don't yet, it's something I would like the team to do, but we don't yet publish specific versions of best practices. We just kind of refer to version of GATK and the website, which I realize, you know, we know that that's not quite sufficient for having something to cite. But the reality is that a lot of people re-implement the workflow anyway. I think the best way to do it is provide the actual scripts or commands or whatever that were used, provide that as part of the methods, and maybe have a short outline that cites the main steps. You know, a little overview to give you a sense. Also for people who don't know what that means. It's a lot of work to ask people to go to a website and read read a lot of GATK documentation to understand what you did to the data in your article. I think having a short summary is a good thing, and then providing the original scripts and commands and all of that is something that should be done systematically as part of the publishing of any computational analysis today.
Well, thank you for that. And we'll, in chapter 14, when we talk about reproducibility, we'll cover that in much greater detail. I saw Matthew give a broad smile when you talked about listing best practices. Matthew, did you want to add anything to the discussion? It sounded like you were gently bemused when she brought that up.
Not bemused, but like that was a guilty face, because I definitely have well, the preprint that we use mentions best practices, and famously it didn't even follow best practices, so that was a big oopsie. So that goes to the point Geraldine was making about writing best practices and actually saying what version of GATK you're using, and if you're actually following every step to the actual letter. So I think that's an example of what not to do.
Well, I would say, and I was not targeting that at you, Mathieu, but it's,
no, I know.
It's often legitimate to make modifications and stuff like that. The goal of the best practices is not to police what people do and say you have to do it this way, it's the only way. I recently saw a discussion on Twitter where somebody was saying that, you know, GATK best practices is the gold standard. It's like, we're not saying that. We're calling it best practices, as in it's the best way that we have found to run GATK for variant discovery in human genomes. And it comes with a lot of specific, like, it's for that use case, which is the use case we apply the most at the Broad. Like, that's the best way we have found to run these tools, which we do know very well. But there are a lot of reasons why you might need to tweak things depending. Now, if you are going to run the exact best practices in that sense, and you're able to run our original whittles that we release, I think that makes it easier for everyone in terms of reproducibility, in terms of also being confident that you can analyze the variants that were produced for multiple data sets, and you can analyze them together, and you know that you're not going to have any batch effects. So differences in variant calls that are actually caused by the methodology as opposed to the underlying biology of the individuals. So the more you can use the exact same things, same processing, even scripts, if you're applying the tools to that same type of use case, that is better. But again, we're aware that a lot of people are doing variant calling on non-humans, including all sorts of organisms, plants, fungi, microbes. Now, there's actually a project in the GATK team, which is called GATK for microbes, I believe. We don't try to be too inventive about names of things. And that is, you know, we're trying to figure out, provide more guidance to people for analysis of non-human and non-model organisms in general. But there's a lot of experimentation involved. So we completely expect that there'll be a lot of variability in how people use the tools, and that's totally fine. We just hope that we can provide enough guidance for people to be successful with that.
Well, thank you. And I'll post a link to the best practices that Mikhail mentioned on the Broad website, the documentation. So we'll put that in the notes for this week. There's a follow-up. So Adelaide posted a question about the best way, getting back to the question about versioning and best practices, best way to capture those versions. And her question was, what do you think about using GitHub and posting a Jupyter notebook with the versions and quote fixes that you use to get where you were? Is this something that we'll cover later in the book, or any thoughts there?
I mean, we'll definitely talk about that to some extent when we introduce the workflows, because ultimately most of these tools and most of these this processing is done through workflows, not command by command. In the book, we do it command by command to kind of get the sense of what's happening when you run a big workflow. But most people will run these things as automated workflows, and there's no equivalent to posting the actual workflow script, because then you can absolutely, if it's a workflow with, the containers, then you have potentially, theoretically, absolute reproducibility of what was done. I like the idea of posting a Jupyter notebook as well, if you want to provide additional insight and commentary on what's happening at each step. I think for educational purposes, that's very useful, and that we actually do that later in the book as well, I think. And we do that in workshops. It's probably not something that we can expect investigators to post systematically as part of their method section, but hey, if people want to do it, I think that definitely adds value for sure.
Very nice. Okay, thank you. Well, we have just a few more minutes before we wrap up. I'll pause there for a final question from our members. Thank you for Adelaide for that one. Last thought.
So I have a question actually.
First meeting. Please.
All right. So regarding this best practice, so is there any like archive of the best practices that evolved throughout the time so far, and then like a list? So what are actually the changes from this best practice to the next best practice? Because I find maybe the best practice are evolving so fast, and then scientists are, wouldn't really transfer to this most recent best practice, like the problem with using the genome builds, reference genomes. So
yeah, I would say we don't have like a systematic archive that says from version to version. I think we will get that. So in the past, the best practices, we didn't share full scripts, which I just said was the best thing to do, but we didn't, because the internal scripts that we used were very specific to a bespoke workflow system that was only used internally. And so that was like the scripts. And gosh, I looked at them at the time. I thought, oh, maybe I can translate this. And there was like a collection of Scala. It was very complex. And even the engineers were like, oh, I don't even know how we would go about translating this into something runnable outside the Broad. When we brought in, when they developed Cromwell and Whittle, that was in part a solution to that problem, because now we can emit them exactly. And so we can actually point you to the exact full scripts probably since 2015, 2016 that were used internally. But there's not systematically a commentary, and the production scripts are fairly complex. So it's not trivial for someone to read through and understand what was changed. However, the team that is responsible for those now they have a new repository that's called WARP, for Whittle Analysis Research Pipelines. I think they really wanted to keep it within the Star Trek universe. And now when they have a new version of the production pipelines, there's like commit messages. It's in GitHub. There's commit messages. There's summaries of what has changed and stuff like that. So I think that situation is going to continue to improve as as certainly our data engineering team kind of ramps up their processes for making things more transparent and reproducible. Yeah. I would say that's a big thing. There is, the caveat is that there is, those are the specific implementations used at the Broad of the best practices, which are technically principles. And so I think on the principle side, we still have some work to do. The GATK team has still some work to do to be a bit more systematic in publishing summaries of when the best practices change, what happens. We will have some of that coming out when we switch for the short variants to the Dragon GATK, because that will come with a couple of major changes to the best practices. And there will be a lot of documentation around that. I think for the big changes, like if we take out indel realignments, as we did a few years ago, when we take out base quality score recalibration, which is going to happen this quarter, those things typically get documented, but it's smaller changes are sometimes not as well tracked, and we'll try to put some effort into that. And hopefully that will help with that problem.
Great. Mikhail, thanks for the question. Great. Okay, well, I want to thank Mikhail for the presentation today. And also a reminder that there's no better way to learn the material than to present it. And if you'd like to volunteer for Chapter 6 or Chapter 7, as we dive deeper into germline and somatic calling, please, you're more than welcome to sign up, or just let us know on the announce channel. And happy to provide templates and everything you need to get going. If we don't have any volunteers, I'll pick up next week's presentation. But Mikhail, again, thank you so much for a very polished and flawless. I mean, there you are in Indonesia, and it just speaks to the global nature of the work that we all do. And I just find it really, really cool. With that, I think we're going to put a pin in it, and we'll call it a day. I want to thank everyone for joining us. Geraldine, thank you for spending some time and answering questions for us, and to our members for their questions. I again, really appreciate everyone's time and perseverance in being here through the holidays. I know many people have taken time off, and thanks for, we're just driving through, and we're going to get this done. So thanks for making an effort. Any last thoughts before we close up? Okay, terrific. Well, again, thanks everyone for joining us. We'll talk to you next week, and continue with this great new year in 2021. Thanks again for being with us today. Bye-bye.
Bye. Thank you.
Bye.
Thank you. Bye.